Hello friends, welcome back to my channel, and today in this video we are going to study about cytokine families and associated receptors. In that today we are going to study about the first family, that is interleukin-1 family. So there are total of 6 cytokine families. Here in this table, as you can see, the family name is given, the representative members of the family are given, and the comments. That means their main function. So the first family is interleukin-1 family, which consists of members 1 alpha, 1 beta, 1 RA, 18 and 33. Next family is hematopoietin class 1 cytokine family. Under this, maximum all the interleukins come under this family. Interleukin 2, 3, 4, 5, 6, 7, 12, growth hormones, everything. Next family is interferon class 2 cytokine family, which mainly consists of interferons and some interleukins like 10, 19, 20, 22 and 24. Tumor necrosis factor, it is easy to remember the name because all with tumor necrosis factor alpha, beta, FAS, BAF. Next is interleukin 17 family, consists of 17, 17A, 17B, C, D and F. And next is chemokines. So the main function for, like here, it is hematopoietin class 1 cytokine family. So these hematopoietin class 1 cytokine family, they are responsible for proliferation, differentiation, maturations. As the name suggests here, growth hormones, they are responsible for the growth, that is proliferation. Cell growth means in eukaryotic cell, the cell growth is the cell proliferation, increase in the cell number. Next come to the interferons. If we talk about interferons, they are mostly responsible for antiviral responses. So the members of interferons, they mostly function to kill the virus infected cells. Next is tumor necrosis factor. Tumor necrosis factor, as the name suggests, they'll kill the cells that are tumorigenic in nature. Next is the interleukin 17. Interleukin 17, they mostly promote neutrophil accumulation and activation. Chemokines, they are mostly function as a chemoattractant whenever any antigens enter in our body. So the cells present nearby the antigens, they will secrete some chemokines to call other cells of the immune system. So the chemicals they release, these chemicals are chemokines. So from the name itself, we can get a little bit of idea about the function of what these cytokines do. To remember the members of the cytokine, if you can remember that interleukin 1, so if you can see here, it is all 1 alpha, 1 beta, 1 RA. Apart from that, we have only two, that is 18 and 33. For hematopoietin class 1, whatever interleukins you can remember, that is interleukin 2, 3, 4, 5, 6, 7, 12, 13, everything comes under this hematopoietin family or class 1 cytokine only. Interferon, again, it is easy to remember because it is IFN alpha, beta, gamma, correct, and few of interleukins. Similarly, for tumor necrosis factor, interleukin 17 family means you can see here 17A, 17B, C, D, F, all with the name 17. So this is easy to remember. Next comes the chemokines, means those function as an attractant. So for that, you have to remember interleukin 8, CCL 19, 21, like this. So if you can able to remember just a few of interleukins and the overview of their functions also, it will be easy for you to answer in any competitive exam. So now today we will study the first interleukin family, that is interleukin family 1. So what are the general features of these family? So first, these are secreted very early in the immune response. As the name suggests, if you can see interleukin 1, 1 means they are in the first position. So whenever any antigen enters in our body, so these are secreted very early in the immune response. And the main secretors are dendritic cell, monophages and macrophages. And when they secrete interleukin 1, whenever they will recognize any viral infection, parasitic infection, bacterial antigens, they'll secrete. Interleukin 1 has systemic effect, that means the whole body effect. And they also signal the liver to produce acute phase proteins, for example, type 1 interferons, interleukin 6, etc. They induce multiple protective effects, they help in the destruction of viral RNA and generation of systemic fever response, and interleukin 1 activates both T cell and the B cell. So these are the general features of interleukin 1. So you can remember 1 means first. First means they are the first to be secreted by immune system and they are secreted very early in the immune response. And the main secretors you have to remember, that is dendritic cell, monocytes or macrophages. Now, if we study about the cytokines of interleukin 1 family, so for one we have one alpha and one beta. Now these are secreted as precursors. Precursors means they need to be break down into active form. So proteolytic enzyme caspase 1 break these precursors and convert them into the active form. One alpha, this is membrane bound form, whereas one beta is a soluble molecules. Coming to interleukin 18, it is related to interleukin 1 only. Similar receptor family, similar function. Interleukin 33, these are expressed in smooth muscle and in bronchial epithelial. 33, they induce CH2 cytokines that promote T lymphocyte interaction with the B cell mast phase and eosinophils. This 33 is also responsible in case of asthma and inflammatory airway and bowel diseases. 1RA, this is antagonistic ligand of interleukin 1. That means the function of interleukin is reversed due to interleukin 1RA. This is responsible to reverse the effect of interleukin 1. It stops the effect of interleukin 1. So these are the cytokine receptors of interleukin 1 family. As you can see here, interleukin 1. This is interleukin 1. And the receptor for interleukin 1 is, it's a dimer. So it consists of two parts. When this interleukin 1 binds with the receptor, it will generate intracellular signaling. Now the inhibitor of these receptors are their monomeric form. Because the receptor function in a dimeric form, its monomeric form will function as an inhibitor, as well as the molecule that function as an inhibitor is 1RA. Next is interleukin 18. So for interleukin 18 also the receptor is in the dimer form. The inhibitory ligands are these two, the interleukin 1F7 and interleukin 18BP. So these are the inhibitory signals for interleukin 11. Next come interleukin 33. For 33 also the receptors are in the dimer form. So the inhibitory receptors are when they are in the monomeric form. So why these inhibitors are needed? Because so that the effect of interleukin 1 or 18 or 33 should not be continuous in our body. Anything in our body, whenever any process starts in our body, once the function is done, that process needs to be stopped. So these inhibitors help to balance those things. Otherwise, what will happen? Once it is on, it will remain on continuously. If these cytokines are secreting continuously in our body, then they will start harming our own cells only. Right? For example, if any person who is angry at you because you have done something wrong, it is okay. But that anger should get controlled after a particular time. It should calm down. That fellow should calm down after a certain time or after you listened to that fellow. But what if the person remains angry continuously? Whatever you are talking, whatever you are saying, he is responding angrily or he is behaving angrily. So that is not good, right? That will harm you. So for anything, even the anger also, it is good when it is required, not always. So similarly, all the process of our body, be it a cytokine release, be it any particular process, for example, secretion of insulin. So every process needs to start at a required time, and once the function is done, the process needs to be stopped. So for that, the ease inhibitors help to balance that thing. Okay, now coming to the signaling of interleukin 1 receptor. So once interleukin 1 binds with the receptor, this will get activated. All the activation starts with phosphorylation. So this first one, MID8, it is an adapter. Because of this adapter, this IRAK will bind. It will autophosphorylates and phosphorylates other members and activate the TRAF6. What it will do, it will activate the ubiquitin ligase so that ubiquitination can be done, and also it will activate TAK1, TAK associated binding factors. Once these are activated, now it will lead to two pathway. One is MAP kinase cascade, one is NF kappa beta pathway. Through this one MAP kinase pathway, the transcription factor which gets activated is AP1. Through NF kappa beta, the transcription factor which gets activated is NF kappa beta. This pathways we already studied. So it's your homework. Do let me know in the comment section the members of MAP kinase cascade and nuclear factor kappa beta. What are the members of these signaling pathways and their sequence of their activation? So you have to let me know in the comment section. So this is only the same thing. It is written here in a flowchart form. That is, once the ligand bind, the conformational alteration happens in the cytoplasmic domain, due to which the adapter protein binds. Due to binding of adapter protein, IRAK will bind. IRAK will activate TRFK. It will activate lubiquitin ligase, and this will dissociate from TACA1 and TAB1 and 2, and it will get activates. Once it is activated, it will activate IKK complex, and then it will lead to two different pathway, either the MAP kinase cascade pathway or the nuclear factor kappa beta pathway. Now here it is written destruction of L kappa beta. So this we have already learned in the signaling pathways. And finally, what will happen? The upregulation or the expression of the genes in response to this particular interleukin-1. So this is about the first family of cytokines, that is interleukin-1 family. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.